Welcome to our second session today in the Savoy 2 room. We're supposed to be in Christchurch, but we're online. My name is Tim Maifuleni, the Regional Manager for the Northern Region at Leukaemia and Blood Cancer New Zealand. And I'm here with our speakers today, Dr. Ian Morison, who is a hematologist with the Southern Community Laboratories in Dunedin, where he diagnoses all types of blood disorders. He particularly enjoys a difficult diagnostic challenge or coming across a rare condition that he has not seen before. As an academic at the University of Otago, he is in the privileged position of being able to explore some of the puzzles in his research laboratory. His main research interests at present include the genetics of myeloma and of thrombocytopenia. We also have Peter. Dr. Peter Browett is a professor of pathology and head of the Department of Molecular Medicine and Pathology at the University of Auckland School of Medicine, and is also a consultant hematologist at Auckland City Hospital. Peter has had a long association with leukemia and blood cancer as medical director and trustee for many years, and more recently joining LBC's Independent Medical Scientific Committee overseeing research grants. Our first speaker will be Ian, and we'll hand the time over to him.
Great, thanks very much, Tim, and good to see you. In quotes, I'm glad you're all lined up here, and I hope this is working for everybody. So I'm going to start by sharing my screen, and we'll see if we can get underway. Here we are. And I'll just move things. Hopefully, you don't have too much in the way on the screen, and you can see this clearly. So this talk is in two parts. So I'm going to kick off, and then Peter Browett will follow. So we'll have a bit of a changeover of the slides when we get halfway there. So we're going to talk about genes and genetics and blood cancers, a tiny bit about familial blood cancers, but we really didn't want that to be too much the emphasis today. So the word genetics and genes, it's tricky because we use these words in so many different ways. And I suppose if you talk on the street about genes and genetics, the first thing you think about is what runs in the family. Your familial genes, your familial genetics, the heritable stuff, the stuff you inherit from your parents and from their parents before it. And so it's a common way in which the word genetics is used. So we talk about heritable genes. A word that we use a bit more scientifically is the germline. That's the stuff you were born with. Or your constitutional genetics. It's the stuff you have in every cell, and the stuff from your parents. Now, in the blood cancers, it's far more important or more relevant for most of us, the vast majority of those with blood cancers, to think about genes within individual cells. So we talk about genes, the cancer genes or the cancer cell genes. We talk about mutations in those genes. Another word we use quite a bit is somatic genetics. So somatic, the soma is the body. So that refers to changes within the body rather than the things that you were born with. And a lot of what we're going to talk about today, the vast majority of changes that occur in your body, in your bone marrow, leading to the blood cancers. So this is where we start. So a bit of a general introduction about genes. Obviously, we inherit our genes from mum and dad. There's an egg and sperm, obviously, and we get 23 chromosomes from mum and 23 chromosomes from dad. And in those chromosomes, within packaged inside, there are about 20,000 genes. So we get 20,000 genes from mum, 20,000 genes from dad. So we have 2 copies of every gene. Now, those inherited genes, most of those are not related to blood cancers. So for the vast majority, I'm going to stretch a little bit on numbers, maybe 99.5% of people with blood cancers is not related to familial or inherited genes, but they do have a small effect. So the stuff we're born with does have an effect on our risk. So it's a bit of a seesaw, a comparison. For those of us who have got pale skin, for example, there's some pluses and minuses, aren't there? Now, if we've got light-coloured skin, then our vitamin D levels are going to be higher, but we're at higher risk of skin cancer, aren't we? So a lot of genetics is like that. And there's probably some genetics, well, we know there is some, that relates to the blood cancer. So variation in some of the genes that are involved in blood development can have pluses and minuses in the same way. So an example, maybe some people have got a stronger immune system. They've got more cells in their immune system. But probably, and perhaps I've got some ideas about this in myeloma, for example, maybe those people who have a better immune system have more cells and a higher risk of actually developing a proliferation of one of the immune cells. So a bit of a seesaw. So there are lots of, we're born with lots of things that alter our risk of getting a blood cancer, but they're pretty small effects. So back to genes. So we've got 23 chromosomes, we've got 23 from mum, 23 from dad. They came together and formed the first cell in the body, which we call the zygote, and that's got 46 chromosomes, obviously. Now, we can look at those chromosomes, and we do that all the time, or the clever scientists in our lab look at it all the time, and they get a karyotype. So in the middle panel there, you can see 3 cells. 2 cells are intact, and 1 cell at just the right stage of its division has been dropped onto the slide, and the chromosomes have spread apart. It's called a metaphase or a chromosome spread. Now you can take, we used to do it with scissors, take that piece of paper and cut it out with scissors, and you can line all those chromosomes up and to form the panel on the right-hand side, which is called your karyotype. So if you look at, you can find the individual chromosomes and they all do line up. They're exactly the same ones. I'm always amazed at people who can do this, because when I look at it, it just looks like 46 blobs, but they're very clever and they can identify those and identify abnormalities within those. I'm going to talk a little bit about it briefly, but Peter will discuss it in a bit more detail. Now, each of the chromosomes is like a recipe book. And so it's like in every cell in our body, we have a stack of recipe books. Within the chromosomes, within all 23 from mum, 23 from dad, there's about 20,000 genes. And they've got recipes for things like recipes for how to make hemoglobin, how to make all the components of muscle, how to make cells grow faster, how to stop cells growing. So there's recipes for everything. So let's push this recipe analogy a little bit in a minute. These recipe books, you can actually, when the human genome was first sequenced, they printed them all into these huge number of volumes of books, which are essentially our recipe books of our genetic sequence. And here, the book that's open is part of chromosome 6. I figured it out last night. And as you can see within that, the letters of DNA that make the recipe. So that you're actually looking at the recipe for a gene there on chromosome 6. So I'd happen to be down where we are at the moment. I've got, just happen to have Nigella Lawson's How to Be a Domestic Goddess, and in there is a recipe for lemon and almond cake. And it starts with zest and juice of 2 lemons, some butter and sugar and eggs and a few other things. So the same is true as we go for the recipes for our different genes. So we can look up, so I looked up yesterday or day before, the recipe for hemoglobin, and this is what it looks like. ATG, GTG, CAT. So those letters of the DNA mean, or is translated by the cell, take a methionine. Now that's an amino acid. Add a valine, add a histidine, add a leucine, add a tyrosine, add a proline, add a glutamate. All right, another glutamate. So that's the way the recipe goes, and it makes hemoglobin. Now, we start from 1 cell and we end up to be a fine species of human. And the average human has got 37 trillion cells in them. Now, importantly, blood cells make up about 28 trillion. Just shows how important haematology is to us. So that's an awful lot of cell divisions. So starting from 1 cell, we go to 37 trillion, but on the way through life, we're constantly renewing and replacing cells. So cells don't live forever. You know, of course, some of the white blood cells only last for a couple of weeks. So they're constantly being renewed and replaced. For example, in the blood cells every day, we make 250 billion new red blood cells and 30 billion new white blood cells, mainly the neutrophils. Now, as we're doing that, we're taking all those chromosomes, all those genes, and dividing and dividing and dividing. Mistakes happen. Every time a cell divides, it makes mistakes. This is a brand new area of science at the moment. There were 3 papers in Nature last week. Nature's sort of the premier journal, talking and annotating how often mistakes are made every time a cell divides. Imagine you made a mistake if you're Nigella Lawson and you put lemon cake, zest and juice of 2, just 1 letter difference, 2 demons. Oh no, what sort of cake would that be? Well, the same thing can happen in our genes. So here, for example, on the right-hand side is the gene for JAK2. Now, some of you who have, say, polycythemia or essential thrombocythemia may be familiar with JAK2. And this is what the middle of the JAK2 gene looks like. Now, stuff happens, mistakes occur, and somewhere through your life, in one of the stem cells, there was a mutation, just 1 letter changed in JAK2. And instead of JAK2, the recipe saying take a tyrosine, a glycine, a valine, a cysteine, another valine, it changed. That changed from a G to a T, said take a phenylalanine. And that changes the whole function of JAK2. Now, I don't want to talk about the function of genes very much, but I thought I'd just talk about JAK2 just a little bit. Oh, sorry, there's just saying JAK2 mutations occur very commonly in polycythemia vera and pretty commonly in essential thrombocythemia and primary myelofibrosis. And JAK2 is a bit like the stuff in the back of your light switch. So we normally switch cells on and off with things like erythropoietin and thrombopoietin to say, start growing, or when they're not there, stop growing. But imagine in the back of your light switch, you have something that's always turned on, and no matter what you did with the switch, it's always on. Here's a cartoon that someone did, a normal JAK2, making a few blood cells out of that black box, white box, but a mutated JAK2, it's just spitting out lots of extra cells all of the time because the switch is always on. So this is one example of a way a mutation, a single letter mutation, can give rise to a cancer or contribute. So these mutations are acquired. We call them somatic mutations, is that word I used before, and they're acquired in the bone marrow, usually in the blood stem cells. The example I've just given is just a spelling mistake. A G went to a T, just like the L of lemon went to a D of a demon. Sometimes it's not just a spelling mistake. Sometimes a few letters of DNA are deleted, or sometimes they are inserted. And a common example would be FLT3, which is a very common mutation in acute myeloid leukemia. And there's called a FLT3 internal tandem deletion that's very common. The other big type of mutation is when chromosomes cross over and get tangled up. And so I'd like to spend a few slides explaining this. Now, in every cell in our body, we've got 46 chromosomes. They're not neatly packaged like this. They're like spaghetti all over the place inside the cell. And sometimes, not very often, but sometimes they get tangled up and things cross over. So we call this a translocation. And you can see in this particular example, it's not a haematology example, but it shows it quite nicely, that chromosome 4 and chromosome 20 got tangled up. So part of chromosome 20 went on the bottom of chromosome 4 here, and part of chromosome 20 went onto the bottom of chromosome 4. So we call this a translocation between 4 and 20. Now, some of the time, this translocation causes something seriously to go wrong. And the famous example The most famous example is called the Philadelphia chromosome. It's famous because it was the first abnormal chromosome to be detected. And of course, it was detected in haematology. And it's a chromosome which causes chronic myeloid leukemia. So if we look on the panel, we've got chromosome 9, chromosome 22. And right at two really important genes, there's a crossover. So part of 22 goes on to 9, and part of the bottom of chromosome 9 goes on to chromosome 22. And this is called, the little 22 is called the Philadelphia chromosome. But what it does is it breaks right in the middle of these two genes and makes a brand new gene. So it's a new recipe. It's like, I don't know, what would you do if you combined the recipe for a lemon cake with a recipe for scones or something? It just could be a disaster, or things could go quite crazy. And what happens is you've got half of one recipe and half of another recipe, and it actually makes, the recipe works. It makes a protein, which is called BCR-ABL, BCR-ABL, we call it. And this actually makes a protein. It makes a protein that's called a tyrosine kinase. So this is the fancy word of a protein that adds phosphate groups onto proteins. But this is not a normal protein, and it leads to abnormal cell growth, excessive cell growth, and in the way that it leads to it, it leads to chronic myeloid leukemia. So people with chronic myeloid leukemia are very special because they are the only people that have this particular protein. The fact that they're special is exciting because there are drugs that will specifically inhibit that. And the famous drug imatinib, and there's lots of related drugs now that are used, works very well to inhibit that completely novel protein that comes from the translocation. Now, there are many different types of translocations. This is a picture from Wikipedia and looks at translocations between different chromosomes and the different diseases that they cause. Most of the examples are from haematology, but they're not all, as you can see. There's one fascinating, between 1 and 11 is related to schizophrenia, which I didn't know about. So many of you will have translocations will have been detected as part of the workup of your blood cancer. We use these a lot in myeloma. We use them in some of the lymphomas for diagnosis. Chronic myeloid leukemia is critical for diagnosis and for monitoring treatment. We use it in the acute leukemias, acute lymphoblastic and acute myeloid leukemia, and Peter will talk about that more. So most of blood cancers, there's a mutation that occurs in a stem cell in the bone marrow. Most of the time, those cells die. They're not successful. Mutations are happening all of the time. But maybe once in a lifetime, or maybe once in a lifetime for 20% of people, not so many people, then that clone has some sort of growth advantage. Maybe it grows faster. Maybe it doesn't die when it should die. And so that one cancer cell in the bone marrow, so the green cancer cell in the bone marrow, proliferates and acquires additional mutations. So I've just talked about single mutations, but as the cancer or the leukemia or the blood cancer develops, usually there's additional mutations. And again, Peter will talk about those. So for each blood cancer in a patient, the identification of those translocations and those mutations are often, not always, but often important to allow an accurate diagnosis. So we use them a lot. They will affect treatment decisions. They can identify potential treatments that put you in a certain group as opposed to others that would work with other treatments. And they can provide prognostic information. So this whole idea of we're using more and more information of the mutations and translocations to lead to what we call personalized hematology or precision medicine or something like that. So that's where I'm going to leave it. Questions will come at the end, and I'm going to hand over to Peter. I'll stop sharing and Peter will pick up from here and talk about some of the work he's doing. With a bit of luck. There we are.
No, I'm here. I'm just.
That's great.
All right. So, thank you, Ian, for that, really setting the scene around describing the chromosomes and the genes. So what I want to do is move on to the next step, is look, well, how do we move that into the clinic? Because as Ian sort of said, it's this era of personalized hematology. And I think we've seen the theme in the myeloma talking around the genes and translocation involved in myeloma, and the CLL, chronic myeloid leukemia, and the lymphomas, is that more and more where, although we treat a leukemia in a particular way, we realize some patients do really well, whereas other patients have a different course. And I think we're realizing that there's, although these all look the same under the microscope, there's a lot of difference. And we're now starting to delve down and to look at the different genes and those spelling mistakes that occur within critical genes within the blood cells that influence how a particular lymphoma or leukemia or myeloma might behave. And I've just taken this headline from a Time magazine, because if you go, what are the big breakthroughs in cancer and particularly in hematology? One's the immunotherapy and the CAR T cells, and I've heard about that. And the second is really this looking at the genes involved within a leukemia or the and using that information to guide our treatment, and appreciating that not everybody is the same. And we use the term precision medicine or personalized hematology. So I wanted to talk to you and just where we've got to in the field of acute myeloid leukemia, but I think equally it's starting to apply to myeloma, lymphoma, CLL as well. So when we look at acute myeloid leukemia, is that when we see a patient and look at their blood counts, and then we go down to the laboratory and look at the blood and we look at the bone marrow, there's some difference in the leukemia cells, but they look very similar under the microscope. So here on the right, this is a tube of blood. This is from a normal individual. This is from a patient with acute leukemia. This is the layer that's got the leukemia cells in it. In fact, that's where leukemia got its name from. It literally means white blood. And this, if we look under the microscope, these are the leukemia cells. And we tend to treat these patients in the same way, but not all patients go into remission, and some patients go into remission and then come out of remission again. And so what our goal is now is, well, why are these patients different? What information can we gain from particularly looking at the genes so that we identify and we give the right treatment to the right patient at the right time? And Ian used the example of chronic myeloid leukemia with the Philadelphia chromosome, where we use the drug imatinib because those patients have got the Philadelphia chromosome. It makes the abnormal protein that drives the leukemia, so we inhibit it. So the imatinib's not going to work in a patient that's got a different sort of leukemia that doesn't have that chromosome and doesn't have that abnormal sort of protein. And that's where we're trying to move our field to. So we started off with this, and over the last decade or more, we've routinely done chromosome studies and FISH studies on the leukemia cells, and Ian explained that. So this is where all the genes are sort of packaged, and like Ian, I remain continually impressed and amazed by the clever scientists in our cytogenetic labs who look at your bone marrow. So they grow those cells up in the laboratory. They then, when they're at the right stage, when the chromosomes are ready to be seen, they drop the cell and split it so they can see all the chromosomes. Their computers then separate out the chromosomes, but they analyze them, and they've got these amazing sort of skills, and they'll look at 20 to 30 of your cells. And so that's why, if you think, why is it taking so long for the chromosome result to come back? It takes a scientist 1 to 2 days just to look at all your sort of chromosomes. But here's a chromosome from a patient of ours with acute myeloid leukemia, and this is good. They've got arrows that point out the abnormalities, but here's the 2 chromosome 8. So one's come from the mum, one from the dad. But this one, a bit of this chromosome 8 has gone and is now sitting on chromosome 21. It's sitting there. And although it's harder to see, this clear bit of 21 that was sitting here is gone here. So this is a translocation, and that's what that means, the T. And so it's swapped around that genetic material. And you can imagine, okay, if I've swapped around these bits of DNA and the genes there, that can't be good for the blood cells where that's occurring. And that's one of the drivers for the leukemia. I just showed you this. This just came through our lab 2 days ago. So this is a new patient in Auckland with acute myeloid leukemia. And this is what we call hyperdiploid. So within the leukemia cells, it's actually double the number of sort of chromosomes. And again, I know exactly which of our scientists would have analyzed this, because she's just got incredible sort of skills. So this is involved in the driving of the leukemia. So those chromosome changes give us important information. And so what we're looking at here is, as clinicians, when we're treating patients with leukemia, we collect all the data and we look at how different patients with different chromosome changes, how they've done, how long they've gone into remission, how long they've stayed in remission. And so this is looking at the probability of a patient staying of their survival with acute leukemia with these different chromosome abnormalities. And you can see this is years from diagnosis. You can see some of these are very good risks. So patients with inversion, that's a swapping around of the chromosome, those patients do very well, have a very good prognosis with chemotherapy. Whereas these patients with an inversion of chromosome 3, they don't do so well. And so we want to look at, should this patient have a transplant early on in their treatment, or some more novel or experimental sort of treatment. So this helps guide us and treat patients in a different way depending on their chromosomes. But as Ian said, looking at the chromosomes it's like looking at your shelf with all the recipe books, or it's like looking at all of your books, is that you can see someone's taken the Nigella Lawson book and they haven't put it back, it's not there. And that's not going to be good, because it means I can't make Ian's lemon tart. But the book might be there, but it might have a mistake in the recipe that would result in an error. And so the chromosome studies are useful, but they only pick up whether the book's missing or it's been put back in the wrong bit of the library. It doesn't tell us when you open it. Someone could have ripped out 20 pages of the book. It doesn't tell us that. Or there could just be one little spelling mistake, like Ian's example of the lemon to a demon, and that's critical, but you don't know that, because all you look and you see, oh yes, the book's on the bookshelf, that's good. In addition, about half of our patients, when we look at the chromosomes, they're normal. So those patients, what's driving their leukemia? How do we guide recommendations around treatment? And it's because when we understand, when we're using the book analogy, is that when we look at the genetic changes in the leukemia, and most of these are what Ian called somatic or acquired changes. So you're not born with them, they're just in the cancer cell, the leukemia cell, is that it's like looking at an iceberg, is that the chromosomes studies look at this bit of information, but in fact, whole lots of the action is below the level of detection. It's that 2/3 that are below the C. And that's what we're starting to learn how to look at in more detail. So around 10 years ago, we started to looking at the genes themselves. And we looked at 3 genes that were involved frequently. They had mutations or spelling mistakes within them, within these 3 critical leukemia genes. So FLT3, and Ian mentioned that one, another one called NPM1, which is it makes a shuttling protein in the nucleus, and CBP alpha. And we tested those either hotspot testing, which is where we just look at the area where we know the mutations are going to occur, or if there's a whole lot of different mutations, we sequence through the whole gene. And that was useful, but we're really only looking at 3 genes. And as you saw on Ian's slide, we have 20,000 genes. So that's like going into our recipe book, and we might look at the recipe for lemon tart and see the spelling mistake in the lemon tart recipe. But there's a lot of other books on the shelves that could have spelling mistakes, but we're not looking at those. But it's still useful sort of information. So what's changed? Well, technology has changed. So when I started doing research and I was doing PCR, in fact, I did it manually. I had 3 water baths, and I would sit there sort of moving things around the water baths to amplify the bits of DNA. And when we were sequencing, we could look at between 200 and 600 sort of bases. And that's why Ian showed you the sequencing of the human genome, where they wrote all that information down. That took 10 years of work, the sequencing of the first human genome, and sort of $1 billion in costs. Technology has moved, and this is an instrument that we have in our lab. This is the NextSec 500. So this is about the size of a small oven, and this is able to analyze this many bases. So those individual letters that were in all of those books that were on the shelf. And so we now have the potential to analyze a human genome, all of those 20,000 genes, to analyze that all in over 24 hours. It takes us a lot longer to actually process and look at all the data and figure out what it means. That takes a long time. But the new technology allows us to do that. And to put that in perspective, sorry, I meant to introduce, this is Stefan Bolander, who's Professor of Leukemia Research in our laboratory, who has set up a lot of our molecular testing. And Stefan's sort of German, so this is his slide, that I think this is a German motor vehicle. But the difference between what we used to be able to do of looking just at the chromosomes and looking at single genes was the original sort of bicycle through to the Starship Enterprise at warp speed, is that's the development of the technology that allows us to look at all the genes that have relevance within a leukemia cell or lymphoma cell or myeloma cell. And that's the big advance. So what we've done is with support from the LBC, who have funded this project, is using the next generation sequencing technology, or NGS, is developed a panel initially of 70 genes, now 78 genes, that are commonly involved in myeloid blood cancers and acute myeloid leukemia. Some studies, people look at all of the genes, so we call that whole genome sequencing. Some studies just look at the bit of the gene that's important in the coding part. So that was the part that Ian went through, the actual recipe part, because within the gene recipe book, you've got lots of the gene itself, but you've also got lots of junk in between that just spaces out the recipes. What we do in this trial is we didn't want to look at all the genes. We just looked at the ones that are most commonly involved in acute myeloid leukemia. And we developed it based on a gene panel that had been developed by the German Leukemia Study Group, published here. This is the different genes here, and this is the percentage of patients that carried a mutation within that gene. And our goal of the study was to separate out each patient, figure out which genes had mutations or spelling mistakes, and what was the prognostic significance of that. Secondly, like the Philadelphia chromosome, did any of those spelling mistakes allow us to say, this is a mistake that would result in an abnormal protein that we can target with a particular drug or medication? And thirdly, would it give us a marker where we could follow the response to treatment? So for those who are familiar with chronic myeloid leukemia and the Philadelphia translocation, we monitor the BCR-ABL gene as a marker of the leukemia. So the more leukemia, the higher the level of that BCR-ABL is. As patients respond, they get lower and lower levels and sometimes even go negative. So we opened this project up in February 2019. This is just to show you, there's lots of writing here. This is the level of report that we, as well as being a study, we fed the information back to the clinicians in real time, and takes about 4 weeks for the whole, for the analysis or for the processing of the sample, and then interpretation and analysis, and then we would review it at our leukemia multidisciplinary team meetings. So here's an example. This is one of our patients, Jane Fonda, and looked after my colleague, Dr. Doolittle. And here, this patient had 6 different mutations within their leukemia that were thought to be relevant. And we call these drivers. They're the things that are causing the leukemia. So I've just shown this slide. This is when we looked at it just after the first 3 to 4 months, so the first 22 patients. And if you look at this left-hand panel, so across the top are the first 22 patients, and down the side here are the different genes that we looked at. And each yellow dot is a mutation or a spelling mistake within that gene. And what I want to point out from this is that although all of these patients have acute myeloid leukemia and all were treated in the same way, you can see that none of them are the same in terms of their pattern of mutations. So going back to Ian's example of the light switch in JAK2, that the light switch is whether you switch it on or off, there's something in behind it that's keeping it on all the time. This is like going to your electric board within the house and having all the switches off, but in behind it you've got a whole lot of being automatically switched on all the time, but it's different in each patient. As their power board is different in what's switched on or switched off. So it's not surprising then that even though we treat these patients all in the same way, that they behave differently. So on average, patients had around 3 driver mutations, but it ranged from 1 to 7. And this is they were all different families of genes. They all did different things. We've now analysed, and Dr. Lauren Child, who's one of my colleagues, undertook this analysis. So this is just over 100 consecutive acute myeloid leukemia patients where we analysed them and looked at the gene profile. And what this slide shows is this is the number of patients here and the number of mutations. So a small number of patients, we didn't find any mutations, but they all had chromosome changes that were important in driving the leukemia. And you can see one patient had 14 different mutations. Most patients had 3 to 4 mutations. So the mean was just under 3.5 mutations. So these are the driver mutations that are causing the leukemia. This slide shows the pattern of mutations being involved. So along here, here's all the different genes that we looked at, they're all different families, they all do different things. And these are the number of patients who have a mutation. So NPM1, that's one that we look at on our little hotspot panel. And Ian mentioned FLT3, but there's a whole lot of different genes that are commonly mutated in over 10% of our sort of patients. So that was important bit of information. The next question we asked is, well, that's all fine. You've got this technology and you can do that and you can generate information. Did it help patients? Did it change the way we treat the sort of patients? And the answer is yes, and probably more than we had anticipated. So this slide shows on conventional prognostic criteria. So that's based around the white cell count, the type of acute myeloid leukemia, and the chromosome changes, as patients are in different prognostic groups. So a favorable group, so this is a group that likely to be got a high chance of being cured with chemotherapy alone. An adverse group, so patients that we think with chemotherapy alone a high risk of relapse, and we would think about doing a transplant early on or some experimental clinical trial treatment. And then an intermediate risk group, which is where they really don't fit into the good group or the unfavorable sort of group. And this group we struggle with as the clinical team. We go, should we transplant this patient at their best chance of long-term cure, or would we just treat with chemotherapy and reserve the transplant for later? So a third of patients changed their risk groups. Some moved from an intermediate group into a good risk, and most moved from that intermediate group into this group here, where we would be recommending a transplant as the best chance for long-term disease control and cure. And of the third that changed their group, it directly changed the recommendation for treatment in just under 20%. So 1 in 5 patients, on the basis of that information, had a recommendation for a change in their treatment. In addition, is that in half the patients, we found a mutation that would alter a protein or an enzyme that potentially could become a target for treatment. In other words, we could use, like we do in CML, where we treat with imatinib, we could use a drug that was going to work in that subtype of leukemia. Here's an example. So this was a patient who we found a mutation in this gene that makes an enzyme, IDH, isocitrate dehydrogenase, so IDH2 mutations, and they occur in around 10% of patients with acute myeloid leukemia. And this drug has recently been studied and is now in clinical trial. This is a specific IDH2 inhibitor in acitinib. So it's not a cytotoxic drug that kills dividing cells, so good cells and bad cells. It just works on cells that have got a mutation in IDH2. And this is an early study looking at patients with relapsed acute leukemia that have got an IDH2 mutation. And quite remarkably, this drug just on its own, 40% of patients went into remission. And we're about to open a clinical trial that's going to take patients, will test for this mutation. And if they've got it, patients are going to be randomized. Half the patients will have conventional leukemia treatment, and half the patients will have the conventional leukemia treatment plus enasidenib to see whether it improves sort of outcomes. And then the fourth thing we found, was that over 90% of patients had a marker that we could use to follow the response to treatment. And this is the concept of minimal residual disease or measurable residual disease. So classically or conventionally, when we diagnose remission, it's based on looking at the bone marrow and seeing less than 5% sort of leukemia cells, and that's termed a remission. But we know there's still submicroscopic leukemia cells, probably in the order of 10 to the 10 leukemia cells, which is why we give further therapy. If we've got a marker, and the one we use at the moment is MPM1, where we can detect it down to levels of 1 in a million. So it's like in this children's cartoon of Where's Wally? How do we identify that 1 in a million leukemia cells amongst all the normal sort of blood cells? And we use these molecular biomarkers to track, and that's been shown to be very powerful in monitoring patients. And again, sometimes we can use that to monitor patients and decide whether a patient needs further treatment or transplant or no transplant. And so that's the concept of minimal residual disease, of personalizing it for a patient. So this is my final slide, which is looking at really why do we study the genes in acute myeloid leukemia and increasingly in myeloma? And it's to personalize it. It's recognizing that everybody's leukemia is a little different, and the information guides around the diagnosis, confirming that it is acute myeloid leukemia or myeloma or lymphoma. It gives us more information around the prognosis, what's going to happen. It might influence treatment. There might be a new target for sort of treatment, or it may change treatment of a recommendation for a trial or transplant, no transplant. Is the treatment working? The monitoring of residual disease. And as Ian hinted at, a small number of blood cancers, there is an inherited risk. It's not as established as is in, for example, breast cancer or colon cancer. So you're familiar with the Angelina Jolie story around he has a BRCA1 mutation, and her mother and aunt and grandmother all had breast and ovarian cancer, and she was tested for BRCA1. We're not at that point yet, but one of the discoveries with this panel is that around 5% of the patients with acute myeloid leukemia do have an inherited risk in this newly discovered gene, only identified in 2015, called DDX41. And so it's an area, my message here would be, it's a small number of patients. It's not the norm for most of the blood cancers, but perhaps we are seeing a little more familial predisposition than we had initially anticipated. And so I think to watch this space. So I'll stop now, and happy to take any sort of questions. Can I just thank LBC who have supported this work, and my group, my colleagues in the lab, and also for any of you who have been involved, all the patients who have their samples have come through for analysis.
Awesome, thanks, Peter. I'm just trying to get Ian back on. There we go. All right. There wasn't any questions that that popped up in the question and answer box, and the questions that I wanted to ask have already been answered in your presentation, especially the finding of that new gene panel DDX41, which is really exciting and helpful for our patients, because a lot of our patients are a little bit worried that their cancers are passed down through their family, and at least if we have them coming along to this talk, then they can understand that that might not be the case.
So if I could, so I think for the majority of patients with acute leukemia or myeloma or lymphoma, is that although we are looking for these gene mutations, we're looking at their chromosomes and their FISH studies, is they're all acquired. They're just in the blood cancer cells. They're not in any other cells in the body. They don't have a risk of passing that on to their family members. This was an unexpected finding, the DDX41, and why I think it's unexpected is that it tends to be patients are presenting, the average age for a patient presenting with a leukemia with a mutation in DDX41 is around 65, 70 years of age. And that's unusual because most inherited predispositions, it's a young age. So it's our pediatric colleagues sort of see the patients, and often part of a syndrome with other abnormalities as well. So Ian's group have done a lot of work looking at patients that have got a low platelet count, and so there's often other features before the development of the leukemia. So this was a little bit different. Yeah.
That's awesome. Well, if there's no other questions, I would just like to say thanks so much for coming on today and sharing your expertise and knowledge, both Ian and Peter. I personally really appreciate you guys. I guess it's kind of like playing chess. You guys have all those different moves that you could potentially do with all those gene panels and things. And it's just finding out which is the best move to do for each of our patients, whether they be a queen, a king, a bishop, a rook, or even a pawn. Really appreciate the knowledge that you shared today.
It's a good analogy. I like the chess analogy. It's one I often use, and chess is sometimes a slow game too, isn't it? And particularly around diagnosis. And so I often talk to patients and say, look, we've just got to wait a moment and see what the opponent's move is. It's like a chess game. So it's a good analogy, I think, for the diagnostic process.
Awesome. Well, I hope you guys have a good lunch that's prepared for you already, waiting to go. And if you guys are happy, you guys can jump on some of our other talks throughout the day.
Tim, is lunch not down in the dining room as it usually is at the forum?
Oh, yeah. I think everyone's got a separate dining room at the moment. Oh,
it might be at the back door. I'll just go and look.
Yeah.
Right.
Okay. See you guys later. All right.
Yeah. See you.
Bye.